Okay, good morning. So,
Morning, sir.
okay, wait, few minutes, wait. That document I cannot share in that in your group. Okay. So, to discuss about the root organization, that is the sixth unit. So, sixth unit is the root development organization of the root apical meristem. Right? Is okay, clear? Is audible?
Audible, sir.
Okay, right. So, here, what is the introduction of root to apex here? The root is constitute the lower portion of the plant axis and it usually develops below the soil surface. Right? So the different development and arrangements of the primary tissues in these two organs are many. So here histogenesis of the epidermis of the root to differ from that of the stem. In the seed plants, the primary xylem of root is exarch, and then in the stem that is endarch. So xylem and phloem strands in the root do not form the common bundle, but they arranged alternately. While in the stem, vascular bundles are collateral, bicollateral, or amphibious. Yesterday we are discussing these vascular bundles. So here the root bears no appendages like stem. So no appendages are there that the comparable to leaves of the stem. Roots are devoid of stomata and their branches originate in the relatively mature tissue of the pericycle. The stem where the branches originate from the apical meristem. So root also possess a root cap and there is no parallel in the stems. So radicle present in the seed consist of root meristem and give rise to the first root on the germination of seed. In gymnosperms and dicotyledons, this root develop into the taproot system. So this one is known as the seminal root, while its branches, the monocotyledon, the first root usually dies and early in the growth, the root system, mature plant consists of adventitious root system. The monocotyledons, the roots are mature, usually adventitious, develop from the stem. So generally, these roots do not develop secondary thickening. A taproot system generally penetrate into the soil more deeply that are the fibrous root system. They are lateral bind more firmly the superficial layer of the soil. So here now we discuss about the root to apical meristem. So that yesterday we are to discuss meristematic tissue means that oval in shape or spherical in shape, dense cytoplasm, then centrally nucleus is there, and less vacuoles are present. These are the meristematic tissue character. Small in size, intercellular space has lock. So this one is the apical meristem characters. So in the root system, the primary root develops from the embryonic radicle. The root has a group of undifferentiated cells at the apex, which consist of the apical meristem of the primary root. The meristematic cells at the apex of primary root are undifferentiated and undergo active divisions. The cells consist of dense cytoplasm and a large nucleus. The tissue of mature root is derived from this apical meristem. So, like that should the apical meristem of root also display many patterns of growth, and there are the exist no constant relationship between the structure of initiating divisions. So the root, unlike the stem, the apical meristem produced the root cap, but even away from the it is since initiate the root cap and there are the occupying the subterminal position. So here the root branches are usually initiated endogenously from the meristematic region. So besides this is the difference. The root apex shows no exchange in the shape and structure as seen in the stems because of the absence of the leaves. So, according to various investigators, that is the central initial. A few initials are root apex of spermatophyta. So, other investigators like Close in 1950 believe that there is a large group of initials in the median region of the root apex. So, although the histogen theory has been abundant for the shoot apex, the structure and growth of the root apex are generally interpreted in the stem of the histogen theory. The apical meristem in the case of the root tips is rather short. In the number of initials range from one to many, and where the initials are more than one, they are arranged either in one row or usually the four rows fairly distinct from one another as univariate strips are appear. So, so see here I show to one diagram. So this is the so this is the central cell and the root cap root apex is the see here this one, right? So we discuss on the basis of the activity and distinctness of the estrogens in the stem. The root also the classified into different types are there. In the lower vascular plants, all the tissues of root are either derived from the single apical cell, example equisetum. Some members polypoideaceae and certain species of Selaginella or many are found. Some several initials are arranged in one tie, as in the member of Marathi AC. So all these plants have the same apical structure, organization, both in the stem and roots. So this is a single apical cell or a single group of initial cells forming the entire root and our origin of root cap from the apical cell. So here the gymnosperms, majority of the gymnosperm, there are two groups. Apical initiating cells are one forming the central cylinder, other giving rise to the cortex and root cap. So here the outstanding feature in these cases, the formation of zone from the cortex of the root and formation of the endodermis from the innermost layer of the cortex. The most of the angiosperms, the groups or initials occur in the root apex. In dicotyledons, the terminal group forms the dermatogen and root cap. The median one develops the cortex. So in the upper one, give rise to the central center of the root. So in this type, both the dermatogen and root cap have also same in the origin, and the cap may be attributed to as a specialized epidermis or dermatogen. In some dicots like Ranalian families, Amentiferae and Ranunculaceae, the certain member of some Leguminosae family members also, some of the monocots, all the part of the root appear to have the arise from common initial groups. The epidermis eventually differentiating from the cortex and the caliprogen differentiating from the lower part, and these form the zonation in the root tip, recalls the condition encountering the Marathi AC in the certain monocots like ZMAs outermost initial develop the caliprogen or root crop. So root cap, the median ones, dermatogen and peribulum, and inner ones, plerome, which is the condition generally seen in the angiosperms. So some variations are occur in the monocots. So there are the three groups of root apis found in the dicots. They are reduced to means recorded as the alien sativum, helianthus, and Giaphairanthus. These the first two type of root apex meristem is represented by the single layer and also recorded from the Eucharis, Amaryllis, and Severia, and in the certain conditions all in the allium sativum. So commonly in most of the monocots, the outermost layer of initials the produces the cap, the median one dermatogen and peribulum, and innermost plerome. The root cap, dermatogen, and cortex arise from the independent set of initials are recorded by the Defende in 1960. So which features are reported to back by the Rube in 1876. So in these cases where the root cap origin is independent in the term as a defined the meristem, that one we call as the caliprogen. So rarely in the condition certain members of monocots like Pistia and uh, Hydrocharis, their origin the caliprogen is from an independent meristem which form both the initiating layers. So in the Despande in 1960, he has recognized the three types of the root apex in the monocots. Despite they are recognized the three root apices in the monocots. So the root cap first one, the root cap organized independently and cannot be recognized into the columella and peripheral zone. So this condition has been seen in the certain members like Polygonatum and Sensaria and Ruscus. These some genus. The cap is recognized into columella and peripheral zone. Both is representing independent estrogens. In this condition, the peripheral region of the root cap may be independent of dermatogen and the rest of the tissues of the root tip originates from the separate initials as is found in the case at the he- Himanthus coccineum. In the Agapathus exerinum, the part of the root is originated from the dermatogen and peripheral layer may be free. The root cap is not free from the other tissues of the root apex and there is no distinction of columel and peripheral layers. So these Clovers in 1956 while studying the root tip of Ziamase which is recognized the cap-like structures are region placed between the root cap and active meristem. That one we call as a quiescent center. So here already this one. This is the quiescent center. Right? Mm. So that is between the root cap and meristem. That one we call as a quiescent center. So after is the Despande also recognize such type of quiescent center in the root apices of some members like Cheliaceae and Amaryllidaceae. So all the functions of the quiescent layer is not thoroughly understood. So Close is of the opinion that the center became active at the time of the formation of the lateral roots and when the root tip is injured. It has been observed by the Jensen in 1966. The quiescent center act as rivers of zonal cells, which are the less sensitive to damage, and they became active while the other part of root apex is inactive. So, according to Despande, the meristematic cells below the quiescent center form the root cap, while the initiating the cells above it is form the other tissue of the root. In the evolutionary significance, from the detailed study of the structure organization of the apices in the several of the plants belonging to different diversified affinities. It has been shown now the visualized the evolutionary progress is perhaps from the single center of the growth to the group 3 are the four centers with progressive increase in the independent of the zones. From the following of description of the different types of the root apice, the origin of the root cap varies in the lower part. The root cap has common origin with all the other part of the root. In gymnosperms, it is the especially distally developed part, the peribulum, and the formation of the dermatogen in this condition is bit delayed and also group away from the root cap. In monocots, although they show the same
Sir, good
pattern of the
morning, sir.
development. Good morning.
Sir, can you please tell me which topic you are teaching, sir?
The root apex sixth unit.
Okay, sir. Thank you, sir.
So here the root apical meristem. So focus on the sixth unit, right? So root apical meristem is the complex structure. To understand the development is better to analyze the simple system. So in this view, Arabidopsis root is one of the ideal system for the study of plant organogenesis. So which is used the molecular genetic approaches. Rapid progress has been made in the identifying some of the pathways to regulating the root development. So the primary root grows mainly by the cell division within the meristematic zone. After newly produced cells elongated roots that have built during the embryogenesis. When cells these leaves meristem, they are the incorporated into the zone of the elongation. Hence, here is elongated longitudinally axis of the root. So cell elongation also contribute to the root growth. So finally, the cell enters the zone of the differentiation where they are the begin to produce the feature with the latter characterized there. So some theories are there. Two theories. What here theories to explain regarding the root apex organization. So different theories are proposed by the different plant anatomists. So these root apex theories are also there. That is the first one here, apical cell theory. So apical cell theory, which is proposed by the Nageli in 1878. So apical cell theory proposed that the single tetrahedral apical cell in the root apice, so which is brings about the growth. Only the single cell apex of the root, that single cell to growth to further division. So it has three cutting phases for giving the rise to the tissues of the body of the entire root. So root cap is derived from its base, but such type of development is not found in the angiosperm. So this is the apical meristem theory. So apical cell theory or apical meristem theory also. So which is proposed by the Nageli. So after estrogen theory. So estrogen theory is proposed by the Einstein. So estrogen theory is proposed by the Einstein in 1968. Put the estrogen theory is proposing the root apex region. There are the three distinct zones or estrogens are found in the root. The dermatogen, that is the dermatogen. First one is the dermatogen and plerome and peribulum. So dermatogen that gives the epidermis and peribulum to gives the cortex tissues and plerome which is to gives rise to the central vascular system. That is the estrogen theories proposed by the Haberland means Einstein. So after Haberland in 1914, he is proposed the that protoderm hermy is proposed. So ground meristem and precambium of these three estrogens. Protoderm, ground meristem, and procambium. These three terms proposed by the Haberland. So another theory is the Carper-Kappa theory. So Carper-Kappa theory, that is the FIP in 1917, that is proposed the root apex divide in the two planes. The zone with the inverted T type of divisions were referred to as a Carper, like that cap-like structure, and other with the straight T as Kappa means body. So the theory basically recalls the Tunica corpus theory concept of shoot apex and similarly. So it explains the differentiation behavior of the plants and the species. So another topic here, organization of the root apical meristem. So a few minutes, we wait, I will come. So thank you for waiting. So coming here, organization of the root apical meristem. So here the development zones. The primary root growth occurs at the root apical meristem. The growing root tip of Arabidopsis is organized into a series of concentric cell layers comprising the an outer epidermis and inner cortical and endodermal pericycle layers surrounding the vascular tissues in the middle of the root. In these layers, the number and arrangements of cell is invariant. The root tip is also divided into several morphological zones along the longitudinal axis. So first one, that is the most distal part of the root tip is the root cap. This root cap protect the root as it is pursued through into the soil. So next one, it is the zone of root apical meristem. This is known also zone of the cell division. Behind the root apical meristem is a zone of elongation. So, which is the cell division cases, and there is the rapid cell growth by the elongation. So, lastly, the zone of differentiation, where the cell assumes their final fate. The cell in this zone are produced by the root hairs. That is the differentiation zone. So, coming to the structure of root apical meristem in the dicotyledonous plants, have two organization schemes are there. That is the closed and open systems are there. So these schemes are commonly believed to remain the unchanged during the growth of the root. In many roots, meristems, self life can be traced as a small group of initials. So often these initials are organized near tiered arrangement with the each tier give rise to the particular tissues. So in the radish, the tier one produce the root cap and epidermis, two cortex, and tier three vascular tissues. So in some maize plants, tier one produce only root cap, tier two produce epidermis and cortex, while tier three produce a vascular bundles. So in some other plants shows no tiered
Sir, what is
arrangement,
the meaning of tier 1?
means that group of cells, one part, okay, some group of cells, right? So here means three groups are there. So each group to produce the different tissues, right? So here, so the radial pattern consists of concentric arrangements. So there are the different tissues are there. So in the Arabidopsis, the majority of the root apical meristem is made up of the proliferating cells. They are divided into two sub-functional zones are there. So that is they called promeristem. So in this promeristem comprise three tiers of the initials. They give rise to the root. So here lower tier comprise the central group, which is around 12 cells, give rise to the tip of the root cap. Lower tier means lower portion, right? So, so this is the root cap is called as columella. The columella initials, which are surrounded by the a ring of the 16 cells, which is give rise to the lateral root cap and endodermis. In the middle tier of the cells consist of central group of four cells that is we known as the quiescent center. So these quiescent center act to maintain the initials is an undifferentiated state. This is the strategy to ensure the after initial cell divisions. So the quiescent center consists root apical meristem cell. Hence, this is equal to the central zone of the shoot apical meristem. This is surrounded by the ring of the eight endodermal and cortical initials, which give rise to the cortex and endodermis. The cells in the upper tier give rise to the pericycle and vascular bundles. So, in the pericycle is made up of the cells that can initiate the formation of the lateral roots. So, all the cells in the pericycle appear to, which is produced the lateral root initials, but the under normal circumstance, only the pericycle cells nearest the internal xylem poles perform the this function. In the center of the root, the vascular tissue has bilateral symmetry with the xylem on the axis of symmetry and phloem on the both sides in the vascular bundles. So here another So another here that is the specification of cell fates and tissue differentiation in the root to apical meristem. The sulfates and tissue differentiation in the root apical meristem of Arabidopsis depend upon the cell lineage. So cell to cell interaction, the position of the initial cell and regulation of the quiescent center. So cell lineage analysis has shown the organization of the root is generated by the stereotypical series of the cell divisions in the promeristem. Here, all these pattern of the cell division are invariant. So here, these root apical meristem of Arabidopsis may be invariant cell lineage. So this does not mean that the fate of each cell is determined from the outset of the development. So the if we study the root apical meristem of Arabidopsis indicate the portion rather than the lineage in the important factor of the determining the cell fate. So example, the cortical or endodermal initial cells is the secondary tier of the promeristem. They normally give rise to the daughter cells that divide to produce cells of the cortex and endodermis. If the second one, if the cortical endodermis initials is ablated with the lateral uh, laser beam, the adjustment pericycle initial undergoes to the unexpected division and replace it. So here, the quiescent center also regulate the differentiate of neighboring cells. If one or two quiescent cells were ablated and columella initials that were no longer in the direct contact with the quiescent cells. So similarly, the cortical initials that were no longer in the direct in contact with the quiescent cells. So this is the, so next coming to the some so coming to the this one is the quiescent center. Next one is the quiescent center. This one is maybe asking the short questions. So the middle tier of the cells in the root apical meristem comprise the central group of four large non-dividing cells. That one we call as a quiescent center. So quiescent cells have the characteristic structure that quiescent center maintains the initials in an undifferentiated state. This strategy ensures that the after each initial divisions, one daughter cell is disconnected from the quiescent center and allowed to differentiate it. So close in 1958,
Sir, what is the meaning of the word quiescent?
quiescent center means that is the in the uh, above the root cap. So this is very active portion. Quiescent center is a technical term which is to very active portion in the above the root cap, right? So see here, this diagram is already. So this white in color above the root cap is there. That is the quiescent center,
What is
very
the
active
function
zone.
of quiescent center, sir?
So one. So this one to produce the some amino acids and divisions also, cell divisions also. They are rich in the cell organelles. Okay, protein synthesis also there, right? So, so in the close 1958, that is the autoradiographic studies of DNA synthesis in the root of the GMS. So is noted the central complex region, the a complex region that cells is had few mitochondria and endoplasmic reticulum, very small nuclei, and the low rate of the DNA and protein synthesis. So that is inactive region called as the quiescent center, right? So since such region have been is reported in many plants, so the cells of quiescent center do not divide but maybe become active when the active initials are damaged condition. This one is inactive, but when it is activated, when the active initials are damaged, means meristematic tissue, if they damaged in condition. So this region is act as a reservoir of the initial cells producing the cells, right? Reservoir of initial cells producing the cells. A ring of the 8 cortical endodermal initials that give rise to the cortex and endodermis around the quiescent center. So another some researchers on root apex has shown that the group of cells at the pole of the stele and the cortex, which is have very low mitotic activity. So this group of cells which is appear in the shape of the hemispherical or disc shape, so that one we call as a quiescent center. The concept, the most distal cells in the body of the root divide infrequently, show the little change in the size and have low rate of the synthesis of nucleic acid and protein. So in apex here 1,25,000 to 2,50,000 meristematic cells Quiescent center contain only the 500 to 1,000 cells. So in the various views, as expressed on the possible cause of the appearance of Quiescent center in the roots, one view suggested that the control by the hormones which may be synthesized in the quiescent center, as substances may be stimulate division at low concentration and inhibit at high concentration. Another view in this competition between the cell of supplies the nutrition and hormones. It was also suggested the root cap control the quiescent center in the particular portion of the meristem. One of the regulator of growth in plants is the pressure. The pressure expected by the rapidly dividing the neighboring cells was suggested to cause the inactivity in the quiescent center. So the way file the cells of root tip turn toward the quiescent center also suggests that the pressure extend to the expansion of the temporary initials and their derivatives present in the quiescent center from the expanding thus in the cells from dividing. So another one is the so next one is the root cap. So primary differentiations are there here. Different vascular systems are present. So root cap. So root cap. This one is situated at the tip of the roots, which is protect the root to promeristem and aids the penetration to the growth of the root into the soil.
Sir, what about primary differentiation?
What
Primary differentiation one concept is
means
given, sir.
yes. Primary differentiations means in the root there are the differentiation regions are there here. That is
Okay.
the cortical ground, and vascular means cortex. Okay, Nandi. Primary differentiation, basic differentiations are there. Means here that is the, so that is no important. That's why I skip that one. So differentiation means the cells are differentiated. Meristematic three zones are there. That is epidermic cortex and vascular bundles or vascular cylinder. These are the primary differentiation in any root. Okay, then epidermis, cortex, and vascular bundles or vascular steel. Nothing but we call as a steel. In higher plants, we call as a steel. Epidermis, that is the outermost layer, right? So, so these root cap, the apical meristem is called as a calyptrogen. As a new cells are produced cells on the periphery of the root, right? In Avena plants, in Avena, the time is interval between the origin of root cap cells and it is shading from the cap was calculated to be 6 to 5 days. Environmental condition affected the development of root cap. When plants normally growing in the soil or raised in the water culture, their roots may be fail to develop the root caps. Example, aquatic plants. So aquatic plants they not have the root caps. So root cap has long been regarded as organ controlling the georeaction of the root. When roots normally growing downwards are placed horizontally, subsequent growth in the elongation zones occur along with the curve until the normal downward orientation is situated or attained. So the site of the perception of the gravity in the root cap is the central column of the cells in which is the starch containing the amyloplast act as the statoliths. So that is the gravity sensor. So in the vertical growing root or the amyloplast are sedimented on the distal horizontal wall of the cells in the central column. A few minutes after the root is placed horizontally and amyloplast slides towards the longitudinally walls directly downwards. So within the year 24 hours, the root assumes its normal vertical portion by growing the long curve and the amyloplast return to their previous location next to horizontal wall. See here like this, I show to. So this is so amyloplast, which is the root cap to bend the two downwards. So next one is the epidermis. So the epidermis cells of root are thin walled or usually divide cuticle. The epidermis of root is usually uniseriate. However, in the aerial roots are tropical orchidaceae and epiphytic areas if the epidermis is multiseriate and it is specialized to form the velamen roots. So velamen roots they absorb the moisture. Okay. So epiphytic plants they have the velamen system is there. Velamen roots are present. So the velamen consists of non-living cells compactly arranged and often bearing the secondary wall thickening. in the form of the numerous narrow strips. The velamen is commonly interpreted as the observing the tissue, but some physiological studies on the arcade velamen indicate the principal role in the tissue's mechanism protection and reduce in the loss of the water from the cortex. That is the velamen tissues. In the young root, the epidermis is specialized observing the tissues and usually bear root hairs, which are the tubular extension of the endoepidermis. The region of the root is usually restricted to the 1 or a few centimeters from the root apex, where the elongation has ceased. Root hairs are absent close to the apical meristem and they are usually dies and they dry out the more mature portion of the root. The certain herbivorous plants, especially the water plants, like lack of the root hairs. The plant that is usually grows on the soil, which is produce the root hairs, fail to do they are growing the water. So, so coming to the vascular cylinder. So, coming to the primary vascular tissues or vascular bundles, here are surrounded by the region cells which are termed as the pericycle. The pericycle is commonly one layer of cells in the thickness but may be multiseriate. The pericycle may be composed entirely of the parenchyma. It may contain sclerenchyma. It may be interrupted by the elements of protoxylem. The primordia of the lateral roots in all the spermatophytes and the phylogen and portions of vascular cambium in the dicotyledon development pericycle. So some cytogenous secretory canals occur in the pericycle of some Apiaceae family members. Some secretory glands are appear, means coriander like that. Apiaceae members, coriander, they have the cytogenous glands or canals are appear. So in the roots of certain water plants and parasite, pericycle is absent. So means here that is there is no that root hairs are not present. So xylem forms on the solid core with the ridge-like projection. So see in the radial files of the cells are transaction. So this is I show to another diagram. So this is the vascular bundles are the xylem and phloem. This is the type of vascular bundles are present. Protoxylem and metaxylem. So the primary vascular structure of monocotyledon root is highly variable and often complex. In some, the center is occupied by the single metaxylem vessels. In others, a circular of such vessels surrounds a pith. The number of protoxylem points may exceed 100 in large palm roots.
Sir, what is the
In
meaning
some,
of protoxylem, sir?
protoxylem is the initial xylem. Okay, Nandi.
Okay.
So protoxylem is the initial xylem, metaxylem is the after development xylem. Proto means
Can
first
you please
primary.
explain briefly so that we can understand something, sir? Actually, we don't know.
Okay, okay. Protoxylem, metaxylem, that is the protoxylem means initial one, right? So initial that protoxylem is produced from the that vascular meristem. Okay.
Okay.
Hmm. So another here, okay, continue that one. That is the mycorrhizae. This one is a special
Sir, can
character
you
you
please
can see.
explain that protoxylem and metaxylem with that image, sir?
Sorry, protoxylem, metaxylem.
Yeah, sir. This just now you know, sir. Can you please explain by this image, sir?
Okay, image. So this is the protoxylem. So initial, the starting one is there, na? So this is the protoxylem. So protoxylem that is produced from the vascular tissues means meristematic tissue, vascular meristematic tissue that is produced. Okay,
Sir, the
that
vascular
after.
medicine.
Hmm? Cambium. So that
Vascular.
cambium from that cambium only to produce the procambium. Nothing but we call as the procambium. So procambium to produce the these protoxylem and metaxylem. Okay.
Can you please mark and show us, sir,
So
exactly where
this
is the proto?
you see here. I, so protoxylem this one. So already
Is
that
it
pointed,
possible to mark, sir?
Oh,
Because in our
I
book
don't know.
also, the as image is there, but where exactly that is located, we are not understanding.
Hmm. So small circular like structures are there, na? Xylem. That is the protoxylem. So some large circular structures are metaxylem. Initial protoxylem, that is the initial structure is okay. Wait. Uh, this one is the this is the see here that is the mm,
The down part, sir.
yeah. So that is the below the circular here some this is the protoxylem. This is the metaxylem. Alright.
Okay.
Okay. So the mycorrhizae, another the mycorrhizae is a that is also important. So endodermis and cortex of root, many plants are often associated with the soil fungi. This association between the fungal hyphae and the young roots of higher plants is known as a mycorrhizae. So usually this is the symbiotic both the higher plant and the fungus they are the definite from association means mycorrhizae means the fungi and roots means that myco means Greek word already discussed here. Mycos means mushrooms, rhizae means roots. So that fungus is associated with the roots. That one we call as a mycorrhizae, mycorrhizal roots.
Sir,
Anyone
other
we can.
than this mycorrhizae, can you please tell me other examples for this mutual association, sir, that is symbiosis?
Symbiotic association is also there. Azolla. Azolla anabina,
Oh,
that is association,
okay.
symbiotic association is present. And rhizobium bacteria. Okay, Nandi. Legumes and
Okay.
rhizobium bacteria, that is also symbiotic association. Lichens, algae and fungi,
Okay.
they are formed as a lichens, that is symbiotic association. Right? So similarly here also,
Okay.
that mycorrhizae means that the fungus, they are associated with the roots. So that is the symbiotic relation is also present. That is the micro. So these mycorrhizae classified, that is the fungus to cortical cells of the host, maybe superficial or enter into the cortex. Based on that one, here there are the two types of the mycorrhizae is present: ectomycorrhizae and endomycorrhizae. So there are the ectomycorrhizae in which the fungus envelops the entire root tip with the dense sheath called hyphae mantle and enter into the intercellular space. Ectomycorrhizae means superficially they are formed as a sheath-like structure. So whatever the endomycorrhizae, which is the fungus, forms inconspicuous mantle, but the invades the interior inside the cells, inner side of the cells. They are also enter. That is the endomycorrhizae.
What is this
So that is
inconspicuous, madam?
hmm, మనకు
Inconspicuous.
అంటే కంటిన్యూ కనిపించదు అన్నట్టు. ఓకేనండి,
Okay,
కంటిన్యూగా
invisible.
ఇట్లా క్లియర్ గా కాన్స్పియస్ అంటాం కదా, లైక్ దట్ కాన్స్పియస్
Yes,
గా
sir.
లైక్. So in the ectomycorrhizae, fungus remains predominantly on the outside of the root with a small amount of the penetration between the outer cortical cell. The ectomycorrhizae roots are very short, branched, and appear the stolon. And the surface of the root, the fungal mycelium form the a thick and dense sheath or either mantle. From the sheath of hyphae penetrate into the intercellular space of the epidermis and cortex. No intracellular penetration to the epidermal or cortex cells occur, but an extensive network called as a hunting network-like structures are appeared in these ectomycorrhizae. Hunting
Parting it
or
means what, sir?
network, hunting network-like structures means that hyphae total enter into the epidermis and cortex cells. Only they are occupy the hyphae penetrate into the intercellular space. Intercellular space ante between the cells, gaps of the between cells. That is intercellular space.
This
Intracellular
hearting net.
space అంటే హార్టింగ్ నెట్
Didn't
అంటే మనకు
understand.
ఇప్పుడు ఉమ్, ఒక నెట్ మాదిరిగా ఫామ్ అవుతుంది. ఓకేనండి.
Okay.
Means హార్టింగ్ నెట్ means that is the
Group of what, sir?
group of the cells, like that. All the connected means all hyphae to connected to form a group of cells. ఓకేనా.
Group of cells or group of hyphae, sir?
Hyphae, that hyphae only.
Okay. Group of
Hyphae are
hyphae.
hyphae. That hyphae means all hyphae to formed as a
Okay, sir.
net-like structures. Individual hyphae are together to formed as a net-like structures. Okay, now Horting means number of people or to union of people, like that.
Okay.
A network. Okay, Nandi.
Okay.
So that is the there. And in the endomycorrhizae, a little surface mycelium, but on the extensive network within the root. The endomycorrhizal roots are similar to. So, so endomycorrhizae are divided into two groups. So those involve the septate fungi and those non-involve non-septate fungi. Septate means divisions. Hyphae, they have the some septal-like structures, cell wall-like structures are there. So septate fungi is the rare and forming the associate only with the orchids, right? So orchids are extremely depend on their mycorrhizal association. If the proper fungi were not invaded, and the seedlings would die. So they are the essential for the successful plant growth. So endomycorrhiza occurs in the Orchidaceae families. Orchids. Right. So, so another root nodule formation that is also important here. The roots they have the association with the bacteria that is the benefits to the plants.
Sir, this endomycorrhizae, this one you not explained.
Ade. So that one is also endomycorrhizae involved the non-septate fungi. They are commonly present the most terrestrial plants. Okay, Nani. Terrestrial plants means on the soil plants. So after that penetrate the host producing character, vessels and arbuscules are present. Vascular, arbuscular, mycorrhizae. This one we call as
Sir, what is
vascular,
the use
vesicles
Endomycorrhizae.
of
are.
endomycorrhizae, sir?
Huh?
What is the use of endomycorrhizae?
So endomycorrhizae to observe the observe the purpose phosphorus to give the plants.
Okay,
Mycorrhizae observe the phosphorus to give the plants. So the plant give the shelter to the fungus,
okay.
Sir,
right?
it is
So that is
nitrogen,
the
no, sir.
phosphorus and nitrogen both. Mainly the phosphorus is observed. Phosphorus and nitrogen, but major content phosphorus. So that is a mutually associate. These plants they are give the shelter to the fungus and that fungi to absorb the nutrition means nitrogen, phosphorus to give the plants. That is the uh, mycorrhizae association. Okay, Nani.
Okay,
So
sir.
Sir,
here that
here
is
the
endomycorrhizae.
VAM, sir. Vesicular avascular mycorrhizae. Can you please explain that, sir?
Yes, sir. Yes. So vesicles and arbuscules they are to form the some vesicle-like structures. Arbuscules means fingering-like structures. Vesicles means vesicles, circular-like structures are appeared in the host cells, in the cortical cells. So the fungi enter into the cortical cell. What happened here? Say they are formed vesicle like structures. Arbuscules means they are anchoring or fingering like structures are appear. In the soil fungus extend in a thin mycelium, they are penetrate either into the cell or between the cells. Means cortical cells. When the hyphae contact the that is a suitable root, they do not break either the plasmodesmata and tonoplast of the host while they are extended into the soil, into the cells. Right? The hyphae usually coils extensively, creating the large surface area or produce the contact with the cells. So the other hyphae ramify throughout the cortex, pushing the cells slightly apart. Some of these hyphae swell and forming the vesicle-like structures. So vesicle means swollen-like structures are there. Vesicle, tubular-like structures, nothing but one we call as a swollen cells are there. So that one we call as a VAM, vesicle-like structures. So cortex is one of the hyphae invariant cells, and there is the ramified. The dichotomized, forming the arbuscles. Dichotomous branching-like structures. Okay, that arbuscles are there. They may be the fungus, the hyphae may be divided into dichotomously. They are extended to observe the nutritions. So look like a miniature tree, like that. Arbuscle look like a miniature tree. So the hyphae of the arbuscle fill the phosphorus granules which are observed by the plant. So once the arbuscle has completed its the transfer of phosphate to the plants, it is collapsed and plant cells return to the state similar to the invented cells. So that protoplastids became chromoplasts. They do not accumulate starch as the sugars are being the converted into the fungi. The
Sir,
plants
can you please
also
say
respond.
protoplastids, sir?
Hmm? Protoplastids means
Protoplast.
before. Protoplastids means before plastids are formed.
Okay.
Chromoplasts are there. Different types of the pigments. These are the pigments. Plastids pigments types are there.
Okay.
Okay, Nani. The fungus never penetrates to the endodermis and stay. Only the fungal they can grow on the cortical cells only. All right.
Sir, this endomycorrhiza you
So
said we see in terrestrial plants. What about ectomycorrhiza, sir? Both in aquatic or terrestrial, sir?
no, only terrestrial only, not aquatic.
Is ectomycorrhiza, sir?
Ectomycorrhiza or endomycorrhiza, only the terrestrial plants only they are associated, not aquatic plants.
What is the
Right?
major difference between these two, sir?
Ectomycorrhiza means superficially. Ecto means ecto ante superficial. Endo ante inside the కార్టికల్ మీన్స్ ఇంట్రాసెల్యులార్ స్పేస్ అంటే సెల్ లోపలికి వెళ్తుంది. దిస్ వన్ ఇస్ ఎండోమైకో. ఎండో అంటే ఇన్సైడ్. దట్ ఫంగస్, దట్ హైఫే, దే ఆర్ పెనిట్రేట్ ఇంటూ ది ఇంట్రాసెల్యులార్ స్పేస్ విత్ ఇన్ ద సెల్స్ ఆఫ్ ది కార్టికల్ సెల్స్. ఎక్టో మీన్స్ సూపర్ఫిషియల్, ఓన్లీ ఇంటర్సెల్యులార్ స్పేస్, బిట్వీన్ ద సెల్స్ స్పేస్. ఓకేనండి. ఇంట్రా అంటే విత్ ఇన్ ద సెల్, ఇంటర్ అంటే బిట్వీన్ ద సెల్స్.
Okay.
ఓకే.
Okay.
So that is there. And another one, the coming to the secondary growth. This one is very important in the secondary growth in dicot root. So you can see that
Sir,
one
this one
secondary
I think
growth.
is good, sir. Root nodules, lateral roots,
Root not.
they are still.
Okay, lateral roots. So that is the no need of the lateral roots. Okay,
Why?
that is not important, to skip that one. And the secondary growth in the roots, that is some important differentiation. Means here secondary growth is occur. The cambium then developed as a secondary, increased the thickness. The secondary growth present in monocotyledon rows. The primary xylem and the phloem tissues are arranged in the separate radii in the dicotyledon rows. So pith is absent. When the here secondary growth is occur, the pith is absent. So parenchyma cells present beneath the each phloem patch became the meristematic and form the cambial strips. Here these cambial only to produce the protoxylem and metaxylem. So protoxylem means initial xylem we call as a protoxylem. The number of
Is there
cambial
any
strips are equal.
protoxylem, sir?
Huh?
What is the
Sorry.
function of protoxylem, sir?
So protoxylem to produce the metaxylem. Okay, that is xylem only. Xylem is to conduct the what here water
The
connecting
water models,
vascular
what is the
vascular that vascular bundles they have these cylindrical means they have the vascular bundles. Vascular bundle they have the xylem and phloem. Phloem they to mm, what here water conducting, right? So here the number of Campbell ships is equal to the number of phloem patches are present. For example, tetra root is with four ships of cambium and triarch root with the three ships. So subsequently, the pericycle cells outside the xylem ridges also became the meristematic and joined the cambial strips of formed beneath the phloem groups. The cambial rings, the initially in the form of the very rings.
Sir, voice is not audible, sir.
So, oh, so here the consequence of secondary xylem formation, the primary xylem is pushed to the central part of the root. The primary phloem elements, especially in the sieve elements, get the crushed at this juncture. The root is identified mainly by the radial position of the exarch primary xylem in the central region. So normally the ray initial are differentiated in the cambial, just opposite to the primary xylem. They ultimately produce the broad vascular rays running the across the secondary vascular tissues in the roots. In addition to these, narrowest phloem rays are seen within the phloem tissues. Um, so compensate the ruptured epiblem due to the increase the diameter of the root consequence upon the secondary thickening. Periderm is produced in the outlet tissues of the root. Basically, the periderm of the root is similar to that the stem, right? So here the pericycle cells undergo the periclinal and anticlinal division. The periclinal divisions cause the increase the number of pericycle layer in radial extent. The combined increase in the thickness of the vascular tissues and the pericycle forces of the cortex outwards. The cortex does not undergo increase in this um, circumference, but they became the ruptured and in the cast of together with the epidermis and endodermis. So phylogen rises in the outer part of the pericycle, form the phylum toward the outside. It may be produced the phyloderm toward the inside. But the phyloderm is differentiated distinct the pericycle, which is proliferated before the pathogen was phylogen was initiated. So in the perennial roots, their activity of the vascular cambium continues through the many years. The phylogen also continues its activity, but may become replaced by the phylogens arising at the greater depth in the root. If
the
such
secondary
development,
growth and bypass root? Just to give us some brief explanations, are actually not
so
understanding clearly.
secondary growth, what happened here? That is the protoxylem and metaxylem.
Okay, sir.
Okay, Nandi. So protoxylem, their metaxylem to develop that vascular means that cambium to produce the inside the and outside of the vascular tissues. So that's why they have to extend in their radius. Okay, Nandi. So that is the main character. Means that cambium to rapidly growth inside of the vascular and outside of the vascular.
Secondary growth in root, which is mainly it mainly refers to only xylem and phloem, sir.
Yes, xylem and phloem is extend developed, and here what here they are to develop the phellogen, phelloderm, and here okay, that cambium, vascular
Cambium is what,
cambium to
sir?
continuously to, so that cambium to continuously to develop. Vascular cambium is there na inside the vascular here. Vascular bundles are there. Vascular bundles ఉంటాయి కదమ్మా. So vascular bundles లోపల that is to produce the cambium. Cambium is present. So cambium to produce the tissues. So that continuous it will be produced. So that's why secondary growth is occurred. మనకు లోపల డెవలప్ అవుతుంది కాబట్టి cambium అనేది number of times undergoes the divisions that produced the that is secondary, right?
This phellogen, I understood, sir. This phellogen and phelloderm.
So phellogen and phelloderm is the that is the below the మనకు epidermis, below the epidermis. So that is the protecting layer. After the second growth, that is phelloderm is there. Okay, Nandi.
Okay.
So after secondary growth, what happened here? That is the very phelloderm is the very hard in structure. Phelloderm means pericycle to produce the phelloderm. So that is very hard in structure. After secondary growth, if we touch that roots, that is very hard in structure. Why? Due to the phelloderm is present. Okay, na? Okay, and thank you for attending the class. Thank you so much. Next class we have to discuss another topic. Thank you.
Okay.
Is okay?
Okay.
So in any
Thank you,
difficulties,
sir.
we have to any difficulties we have to discuss in the that practical classes. So don't worry about that one. Okay.
Okay.
Right?
Okay, sir.
Mm.